Welcome everyone to our monthly PARC symposium. As you can see from this introductory slide that I was asked to show, what we are trying to do is bridge pain and addiction science and research and care. Just a reminder that our next PARC meeting is in December, and it will be from Australia in this case, and Elena Bagley will be talking about endogenous opioids, mechanisms up in the brain. And so please put that on your schedule. And it's really my great pleasure to introduce a fabulous scientist and friend from Karolinska in Sweden. Now, Patrik Ernfors, very briefly, because I want to give him the time, earn his PhD at Karolinska. He trained there, but then he went to Boston, where he did postdoc work, but then came back, and he's been at Karolinska for quite a while. He's a member of the Swedish, I don't know if it's called the National Academy, but it's the most recognized membership for scientists in Sweden. And I also saw from his CV, I think it's called a member of the Nobel Assembly, which basically is the one of 50 people who actually picked the Nobel Prizes. And I won't take it personally, but that's okay, Patrik. That's all right. We did okay. But really, it is absolutely a pleasure to have you here. We read your papers all the time. And today you'll be talking about a subject of great interest to me personally and to everybody else. You can read the title, but it's about a molecular characterization of descending control mechanisms in pain and perhaps even itch modulation. Patrik, welcome. Thank you for joining us.
Thank you very much. I'll try to share here.
I stop share first. Okay.
There, do you see it?
Not yet.
Not yet. Just a moment. I was too fast there. There. There we go. It's coming. It's black, right?
Not quite.
Here we go.
There we go. They got it.
Right.
Yep, we got it.
Thanks, Alan. Thanks a lot for inviting me. And it's really a pleasure to be given this opportunity to show you a little bit of our recent work in this field. And I'll focus on our work on descending pain, as Alan mentioned, and how it interacts with ascending pain pathway, nociceptive pathway. And somatization is remarkable. It's equipping us with the ability to sense temperature, pain, touch, itch, proprioception, and all these abilities are actually, or the sensations, the ability to perceive these sensations is due to the existence of different kinds of primary sensory neurons in the dorsal root ganglion. So each of the different types is tuned to respond to different kinds of stimuli. And these DRG neurons project to the spinal cord, where the information is integrated, summarized, and sent to the brain. In this kind of scheme, we have these DRG neurons, so peripheral sensory neurons, that projects to cutaneous tissues and also organs and tissues of the body. And they're relayed in the spinal cord, which project to the brain. And in this representation, it may seem at first sight that somatization is a true representation of the world around us. But is it really that? Or can the same kind of stimuli actually lead to different kinds of perceptions? Is it invariant or not? And I think that maybe the first person that came up with experiments that truly show that it's not invariant, or it is invariant, actually, and that's Henry Beecher. And he was an American anesthesiologist, and he was a military doctor during World War II. And in 1944, he was deployed to Italy, to the village of Anzio. It's just outside of Rome in Italy then, and the soldiers were under heavy bombshell fire for weeks with heavy casualties. And while working at this field hospital, he systematically looked at how much pain the injured soldiers were experiencing and discovered that up to 75% of the wounded soldiers reported no or very little pain, and even refused analgesics. And in that study, published 1946, he concluded that strong emotions can prevent pain. So now we know that psychological and cognitive states can modulate pain sensitivity, such as alertness, expectations, anxiety, depression, long-term stress, sleep deprivation, among many other things, can increase pain sensitivity. And other psychological and emotional and cognitive states such as stress, trauma, placebo, exercise, aggression, and so on can decrease pain sensitivity. So how this is working is basically that there is a descending modulatory pathway that interacts with the ascending pathway. So this is how it works. So you have the ascending pathway here in blue, and then there are descending pathways that can increase pain transduction in the spinal cord. And there are also other descending neurons that can decrease. So basically, when you have an increased descending activity in the pro-nociceptive, more nociceptive signaling is allowed to pass through the spinal cord to the brain. And the reverse, of course, if you have an increase in the inhibitory. And it's believed that this is an adaptive response that is really critical for survival. So we were interested in starting to understand this pathway and decided to start working with morphine to study this, how the descending pathway works. And morphine is a part of, or a constituent of, or rather active ingredient of opium, together with many other active compounds such as codeine, thebaine, noscapine, etc. And opium is extracted from the opium poppy. It's the sap that you see here on the left that is dried. And when you do forensic analysis, this is the contribution of the different compounds to opium. So you can see morphine is the main constituent or active compound in opium. And it's remarkable. It relieves pain, alleviates anxiety, enhances the mood. It's a miracle drug. And if you go back historically in humankind, it's very interesting to see how long we've been using opium. And the Neolithic crop package is defined as humankind starting to live in a fixed place and to domesticate livestock and start agriculturing. And it arises around sometime around 7,000 to 9,000 years ago in the Near East, and then the Neolithic crop package diffuses into Europe around 5,000 to 6,000 years ago. And there are very recent publications actually trying to work out the origin of the opium poppy. And it's these large seeds that are more domesticated. And then, there are also these wild seeds or wild poppies that have smaller seeds. So there has been studies with morphometric examinations of these seeds, and also carbon dating to try to figure out when we really started using opium. And it turns out it's a very long time ago. It actually came with the Neolithic crop package sometime 5,000 to 6,000 years ago to Europe. And domestication, looking at the seed morphometrics, continues throughout, these thousands of years until late Bronze Age, about 1,000 years before the Christian era. when the larger morphologically domesticated seeds are the ones that really dominates. But large seeds spans all the way back here, actually. So there is a domestication, continuous domestication. And there are records of medical use of opium for pain relief and sedation by ancient Greeks and Romans. And it's quite remarkable that morphine remains among the most prescribed medications for acute and chronic pain, or rather opioids. However, there is a dark side to it, as you all know. Analgesic tolerance, addiction, respiratory depression, and overdose-induced death are maybe the worst ones here. So it's very important to try to understand the mechanism of opioids in terms of its analgesic properties. Try to find ways to take advantage of the good parts and get rid of the bad ones. And as you know, it's incredible problems with opioids, and particularly in the US, with more than 80,000 deaths per year due to overdose-induced deaths. Trying to understand how it works based on expression of its receptor, the mu opioid receptor. So that's the receptor that actually is necessary for the analgesic effects of opium, or morphine, opioids, I should say. But the problem is trying to use the receptor as a way to get insights into how it works is very difficult, because it's expressed basically in all different regions of the brain and spinal cord that are involved in processing pain. So this is how opioids converge with our interest in trying to understand this descending pain pathway. Because we thought that we should focus on the rostral ventromedial medulla here. And this is the last relay nucleus or region that projects to the spinal cord and that modulates pain perception in the spinal cord. So basically, if opioids or morphine is working, it doesn't matter where it works up here. Whatever it does that has an analgesic effect should converge onto this rostral ventromedial medulla. And this doesn't come from nowhere. And it's actually a real honor to present here today, because Alan, my host today, already in 1976, worked out the overall anatomical substrate of the descending pain modulatory pathway. That is defined the rostral ventromedial medulla, and that the dorsolateral funiculus projections through spinal cord is the way how the descending pain modulatory pathway works. And incredibly, in the same paper, he also showed that these RVM spinal projections is what confers morphine analgesia. So lesioning the dorsolateral funiculus prevented any analgesic effect of morphine. So we wanted to go back and basically follow up on this study and to establish what the role of the RVM really is using modern technologies, and try to identify the neurons that are important and decipher in the spinal cord exactly how it works. And a little bit more background before I come today to our data is, which I think conceptually is very important. In early 80s, it was found that there are two types of neurons in the rostral ventromedial medulla that have a tonic or bursting activity. There are off cells, and these cells are on until there is a noxious stimuli, such as heat here on the tail of a rat. And then upon noxious stimuli, these off cells turns off, so they become silent. And then there are on cells that are silent, but upon a noxious stimuli, they turn on. So off cells turns off and on cells turns on during following a noxious stimuli. And the interesting thing is that off cells turns off just before the tail flicks in this tail flick experiment. and the on cells turns on just before tail flick. This is very interesting, and this kind of conceptually shaped the field, that there are these on cells that are pro-nociceptive and off cells that are anti-nociceptive, and that these are the ones that tunes the ascending pathway. And it was also interesting, in the 80s, it was shown that morphine decreases on-cell activity and increases off-cell activity. So going back and trying to figure out using single-cell technologies, we thought that this could be helpful to really try to understand the exact cellular basis for morphine antinociception. So the idea was to provide heat and morphine to an animal, dissect out the rostral ventromedial medulla, and regenerate the single-cell RNA reference atlas. So we know exactly which types, molecular types of neurons that are there. And then in the same animal, or other animals actually, capture the neurons that become activated by morphine. And then for capturing neurons, we use immediately early genes. So these genes turns on in neurons that are firing. And so if we drive a Cre recombinase, we can get a fluorescent reporter expressed in the cells that are active. This can also be used to express various chemogenetic tools so that we can manipulate the activity of these neurons. So the idea was to generate this reference atlas, overlay the neurons that become active by morphine, and thereby identifying their molecular identity. And then using the dreads in chemogenetics to link that activity to pain behavior and connectivity. So here is the first experiment we did, and we used this ARC-3 mice, injected dread G, Q, and GI that allows us to either activate or silence neurons that previously were active. So we captured genetically neurons that were activated by heat and morphine, waited 1 week, and then we reactivated or activated and inhibited the previously active neurons in the RVM to see if the RVM alone is sufficient to lead to analgesia. And actually, we are doing this in the same animal. So we can, in the same animal, both activate and inhibit these previously active neurons. So I'll show you just one Frey thresholds here, but we have also looked at nociceptive behavior in these animals. So activation, the heat ensemble, the neurons that become active in the following application of heat to the limb, if we synthetically now reactivate these, the one Frey threshold drops, so they are sensitized. When we inhibit, not much happens. And here's what happens with morphine. If we now activate the previously active morphine neurons, we obtain decreased sensitivity. That is an increased von Frey threshold, where that is actually similar to morphine alone, suggesting that maybe the RVM is the whole story. You know, it can actually confer the same analgesic or antinociceptive effects as morphine just by reactivating the RVM neurons. But then when we inhibited these, there was actually but allodynia in this case. So they become more sensitive than the vehicle, suggesting that these neurons are continuously blocking pain in the spinal cord. And now that we inhibit them, there is increased sensitivity. So the question is now, where is the RVM the whole picture of how morphine is leading to causing analgesia? And to look at that, we gave morphine systemically, and at the same time inhibited the neurons that previously had been captured due to that they were activated by morphine. And while giving morphine and inactivating the previously activated RVM, morphine-activated RVM neurons, morphine had basically no effect, suggesting that the RVM is really, the RVM ensemble is really key to morphine antinociception. So to understand exactly which neurons these are, as I mentioned to you already, we captured the morphine neurons and collected all cells or just cells that became activated by morphine. And we first generated a reference atlas of all cells, found 19 cell types, some glutamatergic, GABAergic, and TPH2, TPH-expressing neurons. And then we overlaid the active cells, and it looks kind of confusing here. It's the red ones, but when we quantify these, we found that two types, GABA9 and GLUT4, actually, maybe I can get rid of that. GABA9 here and GLUT4 decreased in numbers. So these neurons are continuously active, but when we capture them with morphine, activity is decreased because we capture fewer neurons. And then there are two other types, GABA11 and GLUT2 here, which increase in number with morphine.
Patrik, quick question.
Yes.
Are that the same GABA glutamate characterization as you did in the cord?
Pardon, can you repeat?
Is it a same GABA glut populations that you originally described in spinal cord?
No. These are the RVM
They're
neurons.
different. Okay.
Yeah, they're completely different.
Okay.
Very different. So we went on to develop a dual activity-dependent capture neurons, and this is a way where we can actually capture using immediate gene ARC, like explained before with the Cre recombinase, but also crossing this to an animal expressing the TVA receptor under another immediately early gene, FOS, allows us for a dual system to capture active neurons. One that is we can capture with tamoxifen, as I explained before, and the other one using an NVA pseudotyped lentivirus. The NVI binds the TVA receptor that then leads to lentiviral transduction of the neurons, so you can express a package of genes that is packed in this virus. And we use this to understand whether the spinal cord is really essential for morphine antinociception. So we crossed ARC FOS with tomato mice, injected AAV, adeno-associated virus, expressing the GQ activating dread into these, captured the morphine activated neurons, and then we synthetically reactivated these neurons. So now instead of morphine that could act anywhere in the body, we're just reactivating the ones that were activated by morphine in the RVM. And then at the same time capture spinal cord neurons and express an inhibitory dread in the spinal cord. So now we have a system or a mouse where we can activate previously active RVM neurons, and at the same time in the same animal inhibit spinal cord neurons that previously were activated by the RVM neurons. So we do this unilateral. So one side is control in this experiment. And when we look at the sensitivity, we find that in the ipsilateral side, if we inhibit spinal cord while activating the RVM, there is no effect, and it actually drops. Sensitivity increases and the threshold drops, while in the contralateral side, we see a full effect size. So basically on this side, where there is no inhibition, reactivation leads to analgesia or antinociception, but when we block in the spinal cord, the effect is gone. So now what we did next was to determine, is the spinal cord the whole story for RVM-induced antinociception? So we injected morphine systemically, and the only thing we did in this animal was just blocking the spinal cord neurons. And again, you can see the effects here. So morphine really has no effect at all. In fact, the animals become more sensitized when we block, simply because the RVM neurons that normally is continuously modulating or setting the threshold for sensitivity is now inhibited, and morphine really does nothing, while on the contralateral side there is a full effect of morphine. So now we know that the RVM is really key to morphine antinociceptive, and we know that the spinal cord, its projection to the spinal cord, are also absolutely essential. So this is the pathway that is central to morphine antinociception. And to understand which neurons are actually conveying this effect, we retrogradely traced from the spinal cord of the RVM, the RVM projection neurons, plucked them out, and single-cell RNA sequenced and found 6 cell types, whereof 3 of these were also affected by morphine. So basically, as I showed you before, we have 4 cell types whose activity is modulated by morphine. And among these, GLUT4 seems to be an interneuron, or a neuron projecting to places other than spinal cord. And then we have GABA9 neurons that seems to be whose activity decreased by morphine, and that is pro-nociceptive. And we call these transcriptional on cells. And then we have GABA11 and GLUT2, which we call transcriptional off cells that are whose activity is increased by morphine and that are anti-nociceptive. So now the question is, which ones of these neurons are the important ones? And we choose to look at the glutamatergic one. And that was simply because of this projection, that it was one of the main, the neuronal type with the strongest descending projection. So we obtained VGLUT2 mice, inject an AAV FLIPO, retroflippo into the spinal cord. So this virus is transported retrogradely up to the RVM neurons that project to the spinal cord. And then we have a DREAD, GQ or GI, that become expressed only in the glutamatergic neurons because it depends on the CRE as well as the FLIP recombinase. So we get only expression in glutamatergic projection neurons here of these chemogenetic tools. And when we activate this, we have an increase in one-thread, so they're less sensitive in the ipsilateral side of the injection. And now when we inhibit these neurons, as you can see here, we have similar effects as we saw when we inhibited the entire RVM ensemble. And now we're just inhibiting the glutamatergic neurons. And again, morphine had no effect. So these glutamatergic neurons actually express brain-derived neurotrophic factor. And we confirm this in our single-cell atlas when we look. And we confirm this by in situ hybridization. And we also retrogradely traced the glutamatergic neurons and quantified the proportion of glutamatergic neurons that express BDNF. And it was more than 80%. So these neurons are the main glutamatergic RVM spinal projection neurons. And looking at the distribution of these neurons, we see them in the Raphe-Magnus and magnocellular reticular nucleus. So they're kind of widespread here, and they project to the spinal cord, terminating in lamina 1 and lamina 2 outer. So we obtained BDNF2A mice. And now, instead of manipulating the RVM ensemble, all neurons that are actually activated by morphine, or just the glutamatergic, now we are just targeting the neurons expressing BDNF, the GLUT2 neurons in the RVM, a specific cell type. So we express GQ and the activating and the inhibiting DREAD in these neurons. And again, we can replicate what we saw with the entire, when we activated the entire ensemble. And again, inhibition leads to increased sensitivity. And again, if we repeat the experiment we did with the entire ensemble here, we give morphine to the animals, there is a decreased sensitivity. And now if we inhibit these BDNF neurons in the RVM and give systemically morphine to the animals, morphine has no effect. So these BDNF GLUT2 neurons are central to morphine antinociception. And we call these neurons RVM BDNF neurons now, from now on here. So we use these animals, activate the previously activated, we activate the BDNF neurons in the RVM, and in these animals, we collected the spinal cord to find out which kinds of neurons in the spinal cord is necessary for the effects of the BDNF activation. And we found basically just one cell type, and it's this inhibitory 8 cell type that express galanin, so GAL positive neurons. And we independently confirmed that activation of the BDNF neurons in the RVM leads to an increased number of FOS positive neurons in the spinal cord. And these GAL neurons are very interesting. Already in 2018, we did spatial transcriptomics, actually single-cell RNA reference atlas of the spinal cord and spatial transcriptomics. And these GAL neurons are located in lamina 1, 2, mostly in the spinal cord. So they're in the right place in the spinal cord to actually gate pain. So the question now is, how does opioids close the pain gate in the spinal cord? And our data then suggested that these GAL neurons might be critical for this or essential for this. So we obtained a GAL-3 animals and injected an inhibitory dread into the GAL-3 animals. So now we express it only in the galanin positive neurons. So in this experiment, then we inhibit the GAL neurons with and without morphine. And again, as you can see here, these GAL neurons in the spinal cord are essential. And in fact, inhibiting these neurons leads to increased sensitivity, similar to what we saw actually inhibiting the RVM-BDNF neurons. And now if we give morphine systemically to these animals, nothing happens. These animals become as sensitive or even more sensitive than the control, while contralaterally, morphine has its full effects. So GAL neurons are essential. So basically what we found here then is that in the rostral ventromedial medulla, there is a specific type of cells, the RVM-BDNF neurons, that projects to the spinal cord and terminates or contacts monosynaptically. I didn't show you that, the spinal cord GAL neurons. And it is through this interaction that the antinociceptive effects of morphine is elicited. So we were interested in actually what happens here. How does the descending pathway, we knew that it was the GAL neurons, but how is this actually, what is the circuit by which the GAL neurons interact with the ascending pathway?
Can I ask one more question? This is all baseline sensitivity, right? It's a normal mouse. It's not an injured mouse.
No, it's a normal mouse.
Right. Have you looked at it under injury conditions?
Yes, not really injury. The only thing we've done is with carrageenan, and it does reverse carrageenan-induced hyperexcitability or hyperalgesia. But that's about it. We haven't done more there. But I'll get to some other stuff where we have done injury models.
Okay.
So we wanted to understand how it interacted with the ascending pathway. And we had another postdoc in the lab was working on the ascending pathway using the similar strategy, FOS-TBA animals, to trap neurons in the spinal cord that are activated during noxious mechanical or heat stimuli. And he found that in these animals, when he gives a mechanical stimuli, trap the neurons in the spinal cord that become active during noxious mechanical stimuli, and then synthetically, a couple of weeks later, inhibit these. And then he checked then the ability of these mechanically activated neurons, whether there is a cross modality effect or not. So we looked at 2 gram one-Frey nosfensive bevi to 2 gram one-Frey filament, and you can see nosfensive durations really drop when we inhibit these neurons, but there is no effect on heat or cold. And we also trap neurons that are activated by heat, and we had no effect on noxious mechanical behavior, but reduction in nosfensive episodes to hot plate and no effect on cold. And we also, similar to what I showed you for the RVM, sequenced these cells and mapped them onto a reference atlas and found that one cell type, an excitatory 21 cell type, is activated by mechanical noxious mechanical stimuli, and excitatory 19 by heat. And here is that actual data. We generated a new reference atlas of the spinal cord, and here are these inhibitory 8 neurons in this atlas. And here's the quantification where you can see the increase in by heat of excitatory 19 and the increase of excitatory 21 following mechanical stimuli. But what was very interesting here was that both of these stimuli led to a marked increase in capture of these galanin positive neurons. So it seems that there is a modality selective activation of excitatory 19 and 21 neurons, but the inhibitory neurons is activated by both stimuli. So we use these GAL animals for this purpose as well, to understand how these neurons actually are part of the ascending pain pathway. And in this experiment, we expressed an activating dread in these. So we can synthetically activate the galanin neurons in the spinal cord. And you can see that we increase the withdrawal threshold, so sensitivity decreases. And sensitivity also decreases in nose-offensive episodes in the hot place. So both mechanical and noxious mechanical heat stimuli sensitivity drops when we activate these. And the reverse was seen when we inhibit these. So basically, these neurons, unlike the excitatory neurons, are not modality selective. And we confirmed also that they are present in lamina 1 and lamina 2 outer, like we had shown 2018 using spatial transcriptomics. So the question is, how do these neurons...
Could you put that slide back on? It was just on for a brief second. And which is which?
What do you mean, which? The GAL are the red here.
The galanin are the red, okay.
Yeah, and the PKC gamma is the green, so lamina 2 in there.
So it looks like there's some of the gals in 2 and 1.
Yeah, I mean, oh yeah, it's in lamina 1 and lamina 2 outer mostly.
Okay, thank you.
There is some you can see here that in situ it looks like it's actually more in lamina 2 inner and protein. So we have mapped also with single-cell RNA sequencing mouse DRG neurons, and this is a recent paper we had just this year where we found a number of different types and resolved previously unresolved heterogeneity and did spatial transcriptomics and really can map back and identify the different cell type and their organization in the ganglia. And we recently, just a week ago, on bioRxiv, posted a paper together with the U19 pain network in the US, the Precision Network, a joint effort mapping human DRG neurons. And we also did spatial transcriptomics there. So we know exactly what kinds of neurons are there in mouse and human and the relationship. And it's quite interesting if we look at the hierarchical relationship between mouse and human. So all the sensory neurons can be divided into groups: thermoreceptors, anoreceptors, nociceptors, and proreceptors, LTMRs, proprioceptors, C-nociceptors. And all the human and mouse neurons comes together in this organization. And we will look at using machine learning, or rather deep learning strategies, training mouse cell types and then predicting human cell type. You can see it's almost a one-to-one relationship between mouse and human cell types, which is actually really good news for us working on mouse as a model system. So we wanted to understand how these GAL neurons are connected to the ascending system. We used monosynaptic rabies virus tracing. So we traced from the GAL3 neurons, GAL positive neurons in the spinal cord, collected dorsal root ganglion, did in situ hybridization for all these different cell types in the DRG. And they are outlined here now schematically. And as you can see, we're tracing from the GAL3, 65% of DRG neurons belong to what we call the PEP2 population, which is an A fiber nociceptor type. And about 31% or 31% had monosynaptic input from A beta field LTMRs. So these are the two cell types in the dorsal root ganglia that has a direct connectivity, monosynaptic connectivity with the GAL neurons. So it looks like this then, that you have CNA HTMR fibers that connect to excitatory 21 that codes for noxious mechanical stimuli. C heat fibers that connect with excitatory 19 that cause for noxious heat sensation. And within this network, we have A delta nociceptors and beta field LTMRs that terminate on inhibitory 8 that are actually feed-forward inhibitory neurons for the excitatory 19 and 21. And what these RBM BDNF neurons is then impinging on this ascending pathway by tuning activity of this inhibitory feed-forward inhibitory 8 or galanin positive interneuron.
A delta, the A delta population, some of those are also TRPV1 positive, aren't they?
Yes, they are.
Okay.
Yeah, one of them. There are two types actually.
Right. So there might be a heat convergence there as well.
Yes, it might be.
Yeah. Okay.
So what we believe with morphine is that normal physiology is RVM BDNF neurons are continuously active, tuning activity of the spinal cord galanin positive inhibitory neurons that determines the ascending pathway. So therefore, when we inhibit these, gal neurons become when we inhibit these, sorry, I'm getting tired. Yeah, galanin neurons activity drops and the ascending pathway increases. And what morphine essentially does here is impinging on these systems, on the RVM BDNF neurons, increasing the neuronal activity that leads to increased gal neuron activity and decreased output from the spinal cord projection neurons. So let me see here. I maybe have a few more minutes. So we've also looked at the mechanism of sleep deprivation induced hyperalgesia. So sleep deprivation leads to increased pain intensity, decreased pain thresholds, and also worsens pain intensity in chronic pain patients. And this actually happens also in mice, so it can be recapitulated there. And we used a similar strategy here. Capturing the neurons and reactivating them to see if we can recreate hypersensitivity, sleep deprivation-induced hypersensitivity. And here's what it looks in sleep-deprived animals. Sensitivity increased, the threshold drops. It also, withdrawal latency to heat also drops, and a little bit in offensive heat behavior in the hot plate test. And when we capture this and synthetically reactivate only the neurons in the RVM that were active during sleep deprivation, we recreate basically all of these behaviors. So the RVM is also really important for setting the threshold, sensitivity threshold to pain following sleep deprivation. And when we sequence the key, I'm cutting a lot of data here, but the key finding was that one of these GABAergic neurons in the RVM, GABA3 neurons, dropped in activity massively. So we focused on these neurons that express MPY2 receptor. And again, they're kind of scattered throughout the RVM here, as you can see, the red dots. And when we use this MPY2R Cree animal that now only activates this GABAergic neuron, we can recreate this behavior. So the GABA3 neurons or the MPY2R neurons are key to sleep deprivation-induced increased sensitivity. However, when we activate these neurons, we have absolutely no effect at all. Well, it's a little bit here, but the effect is really minor. So we were kind of wandering around this, what's going on here? And then what happened is that when we activate these neurons following sleep deprivation, we can completely reverse the sensation following sleep deprivation. So basically, this suggests that these MPY2R neurons are continuously active, but during sleep deprivation, activity drops. So now that we reactivate them following sleep deprivation, we have an effect size, but not in the rested animals. So here is kind of recapitulating these. I didn't show you that, but these are interneurons in the RVM. So we believe that they project to projection neurons in the RVM that are pro-nociceptive neurons, and that now these are continuously active in the rested situation. So when we activate them synthetically, we have no effect at all. on pain sensitivity. However, following sleep deprivation, activity drops in these neurons, leading to increased output, and these are pronoceptive, so you would enhance out spinal cord ascending pathway here. But now there, because of this, when we reactivate these, as I mentioned, in the sleep-deprived animal, we can now block this and get back to normal baseline sensitivity in the animals. So our idea right now, these are all preliminary data, but our idea now is that these NPY neurons have a decreasing activity with increasing wake time, and the sleep deprivation is just a continuation of this. And if that's true, and it really doesn't have anything to do with the circadian rhythm, then a nap, at the end of sleep deprivation should reverse this. And that's actually what happens here. So we give the animals a short nap, and the baseline is back. So these neurons are actually seem to be a central hub, maybe for chronic pain, but only certain types. So here is a carrageenan injection, and you have a massive drop in the threshold, so increased sensitivity here, and that can be reversed by activating these NPY2/3 inhibitory neurons. And similarly, stress-induced hyperalgesia in the chronic restrained stress model leads to a marked drop in the von Frey threshold. And again, that can be reversed by reactivating these. But what was, I think, very interesting is that in the Sperner-Vinjer model of neuropathic pain, when there is also a massive marked drop in the threshold, so increased sensitivity, but reactivating these doesn't do anything in the SNI model. So it seems to really be essential for normal physiology, in particular during sleep deprivation and during inflammation and stress, but not neuropathic pain. So here are the collaborators. Michael Fatt did most of the work in the morphine story or morphine study. Muge Altenkopf worked on the sleep deprivation, Ming Dong spinal cord ascending pathway, and the human and DRG single cell atlases and studies were stories, Dimitri and Jussi. And we collaborated with Malin in Uppsala, Hans-Ulrich Seilhofer in Zurich, Wen-Chin Lu for human DRG neurons, and Håkan, I didn't show the results on Håkan. As I mentioned, now recently we had a paper last week on bioRxiv with an integrated atlas with over 50,000 neurons as part of NIH Pain Precision Network. Thanks for your attention.
Thank you. I will open it to questions, but I have to ask two questions. You never mentioned 5-HT neurons. I feel so bad for the 5-HT neurons. It's like they don't do anything. But I noticed that the NPY2Rs, many of them are right on the midline where the raphe is. And
yeah,
are any of those serotonergic?
It could be. I mean, quite honestly, we're really struggling with that project because we haven't been able to identify the output neuron. So we're really struggling with that right now. We're trying all kinds of experiments. So we don't know. It could be. We could be.
The other question is, how do you define something? I've always wondered, are there interneurons in the RVM? How are you defining it as an interneuron?
Well, I didn't say interneuron. I said either it's an interneuron or a neuron projecting elsewhere.
Okay, all right, okay.
Because, it could very well be. We don't know.
Okay.
And maybe that's why we don't find it in the RVM, actually.
Okay.
But I think the most parsimonious explanation would be that it would be an interneuron.
Right.
Because, we do see that it's just 6 cell types we actually find, at least with an AAV retrotracing, to be projection neurons. It doesn't mean that there aren't others that we would identify using other methods, but still.
Yeah, well,
go ahead, Howard.
Alan, we never saw any serotonergic neurons that were activated by morphine.
I know.
Yeah, we know that if you microinject morphine into the RVM, you activate off cells, you get inhibition of pain.
Yep.
But that's also blocked by naloxone or an opioid antagonist at the spinal cord level. at inhibition. So do you have any evidence about the location of the opioid receptor in these different neurons that you're doing transcriptomics on?
Yeah, I mean, we don't think that it's the RVM-BDNF neurons that are directly, interacting with morphine. I mean, they could, but I mean,
well, if they're inhibiting pain and they're inhibited by morphine, then you should get hyperalgesia with morphine in the RVM. So logically, you're right. Your thinking is right. But if there's a hyperalgesic cell like the on cell that's supposed to express the mu receptor, I mean, it's hyperpolarized, by mu opioids again. So I'm particularly interested in the Gallin neurons because if you record from cells in 2 outer, and that's been done in primates and in rodents, those cells generally tend to be excited by noxious stimulation. They also, if you go to a spinal cord slice, those cells are hyperpolarized by mu opioids. But I wouldn't expect those cells to be inhibited by opioids, right? If, the inhibition of them makes, pain worse, right? Then opioids on the spinal cord should make pain worse. So it becomes very critical where the mu receptor is.
Yeah, but I mean, I think at least based on our results, it's pretty clear that morphine, the major effects of morphine is, converging onto the RVM-BDNF neurons. But there are effects outside. I didn't point that out, but if we inhibit the RVM-BDNF neurons, or actually more the RVM-BDNF than the spinal gall neurons, we actually do have a residual effect of morphine. We're lifting the threshold from this very allodynic state to baseline, to control line. So, I mean, morphine is acting elsewhere on neurons other than the RVM-BDNF.
Yeah, Tony Yox did some beautiful pharmacological work with injections, RVM and spinal cord, and showed that there's a synergistic interaction if you do a dose response curve.
Yeah.
Right? You need a lower dose in the RVM if you have a small amount of morphine on the spinal cord.
Yeah, I think that's in agreement with our results as well.
It's not in disagreement.
Let me let other people ask a question in case. Are there others who would like? Sorry to interrupt, but anybody else want to ask a question or put it in the chat? Just want to be sure, or else Howard and I will just take over. But I'm going to follow up. I'm going to follow up on one of Howard's, because one of the things we did show, and next time when you show it, thank you for mentioning our paper, you can take the date out. It's not necessary to show the date.
I thought that was interesting. It took so long.
I was only 6 years old at the time.
But you can show the number of citations.
Yeah, no. The question I have is, obviously there's a dose-dependent effect here, because the systemic morphine will, even in a spinalized animal, will work at the cord. And so there's a point at which the descending control system can, if it's inhibited, if you block it the way you did beautifully, a higher dose of morphine should still produce analgesia. And the question is, I'm curious to know, do you see that, or what dose of morphine were you using in these guys?
10 mg per kilogram.
Oh, that's pretty big. Okay, so that's pretty big.
I mean, we never tried different doses, except, I mean, I didn't show you that we knocked down BDNF and overexpressed BDNF. And in the overexpression experiment, we actually dosed up from lower doses up to 10 mg and find a synergistic effect. But I mean, we never did other doses in any of these experiments.
And then I have to ask a question that Frank Parecka would agree with you, but in our hands, certainly with Greg Shearer, we rarely find an effect of morphine or a mu agonist like Damgo on mechanical sensitivity, even after SNI. and clearly you're getting an effect. And I'm very puzzled at that, and I really
Really?
don't
Did you
understand.
give it systemically or
No, that was spinally. Yeah.
spinally? I mean,
Yeah.
that could be the difference.
Yeah, that's
Maybe.
interesting. That's true. That's right. Okay.
Because I mean, what you're looking at is what we call the residual effect.
Oh, harsh. Okay. I'm going to keep track of the dates of your papers. Are there other questions from the audience?
I have one. Hey,
Go
Alan.
ahead.
Yeah. So a lot of the tests that you did were one try, so testing for mechanical thresholds. Have you tried reversing, say, like a CPP, or, do you think that there's actually an action on the brain too, how they're perceiving the pain?
Oh, yeah. I mean, that's a good question. I mean, we in most experiments, I mean, because we did so many different, experimental paradigms here. I mean, we just did Wolfram up and down, but for looking at the RVM ensemble and RVM BDNF neurons, we did look at nociceptive behavior as well, which like licking, guarding, shaking. So I think it does, but I agree with you. I mean, we never looked at aversion itself, for example.
Yeah. And then because you wonder whether activating this population might cause preference too.
Yeah.
Yeah.
What do you mean by preference?
Oh, if you give them morphine and it feels good, you wonder if activating this population.
Okay.
Yeah.
Oh, that would be very interesting. I haven't thought about that, but that would indeed be very, very interesting.
There's a question in the chat from Nicole Laura. Is BDNF administered? This is an interesting question. DeConnick, I don't think he's on the call, but is BDNF administered to spinal cord likely to be clinically useful for analgesia? That's the first part. Second question, which I'm very interested in, is RVM involved in, do you know if it's involved in spinal adrenergic analgesia?
Yeah,
Well,
I don't know why these are coming up under that name. I put those in, Alan Lawrence.
Oh, okay. Anyway,
Yeah.
go.
Related, maybe
No, wait, but
related.
let one at a time. Go ahead,
Oh, I did not.
Patrick. So the
okay.
question
I mean,
was BDNF to the cord, and the second one is, what do you know about in your system? Have you ever used, say, alpha-2 agonist, for example?
no, we haven't. I mean,
Okay.
that I can say directly. We don't know. For the first question, I mean, BDNF is expressed by many neurons in the spinal cord. It's also expressed by some types of primary afferents terminating in spinal cord. I think, BDNF is in many places of the brain and is central for plasticity. And I think that just, giving BDNF might not be the best way of solving this problem, because you would affect, all
Right.
kinds of synaptic contacts and plasticity in all kinds of neurons. It needs to be controlled so that you're only activating plasticity in the ones that are important for what you're looking to change, So I doubt that would be a feasible strategy forward.
There's a question from Anastasia Keller. Thank you for a great talk. I concur. She writes, I am curious, how would electrical spinal cord stimulation, so SCS, affect the RVM anti-nociceptive, pro-nociceptive pathway in the context of chronic pain? That's a tough one.
Yeah, I don't know exactly what you mean by electrical spinal cord. You mean electrically activating the primary afferents projecting to spinal cord?
Hi. Yeah, so this is Nastasia. Spinal cord stimulation is epidural spinal cord stimulation is commonly used to treat chronic low back pain, for example, chronic refractory low back pain, to other treatments. And so in that case, I mean, the leads are placed epidurally. I think they overlay the dorsal roots. So yeah,
Yeah, then you would actually activate primary afferents, I believe.
Yeah, most people think that, at least with SCS, that you might be getting posterior column, dorsal column stimulation, mostly large fibers, but nobody really knows the mechanism. That's just not clear.
Well, I mean, I think, the discovery of the on and off cells has really been important. And I mean, it's pretty clear that there is a direct connectivity between the ascending and descending pathway. And it goes via the parabrachial nucleus that projects. There is, spinal neurons terminate in mouse. Most spinal projection neurons terminates in the parabrachial. And the parabrachial nucleus neurons project to the RVM. So this is a closed circuit. So if you affect, the ascending pathway, you're going to impact on the descending pathway. So I would say that it is involved, and exactly how I don't think it's known. But there is a direct connectivity there between the ascending and descending pathways.
We'll let one more question, and then I'll let you go. It's late. It's 10 o'clock in Sweden. From Izzy Triana, who asked just about male-female differences. You did mention it a little bit, I think. Do you notice any particular sex differences?
No, we did not. Not in these experiments.
Okay.
None of these. Actually, we checked male and female in both for sleep deprivation and the morphine study.
That's
And we
great.
did not see any difference.
Well, on behalf of PARC, Patrik, thank you for a wonderful talk. We got a lot to talk about for sure.
Well, thank you.
We'll see you. You're going to be in San Diego?
No.
Oh, too bad.
I'll be in Verona.
In Verona, d'accord.
Yeah,
Okay.
thank you for, this was a very illuminating talk. Thank you. And
Well,
this is
thank
a
you so
marvelous amount of work. And just the kappa receptor is in a different group of cells than the mu receptor. And I don't know what dynorphin and the kappa receptor are doing in the spinal cord or in the RVM. But you can have either hyperalgesia or analgesia with a kappa agonist in the RVM. So I don't know what's going on. But Patrick's going to figure it out.
Yeah,
maybe.
It's very reassuring.
Thank you.
Well, it's been
everybody.
Yeah, and thank you all. I would just want to say I really enjoyed presenting and having the opportunity, and also meet you, Howard.
Yeah, likewise.
All right, take care.